Okay, chapter 12. I just put this general outline in there. What we're going to look at in chapter 12 is reactions that involve pi bonds. I mean, we've touched on them occasionally up to this point, but they were sort of hit or miss, and they entered the conversation as actually forming the bonds, or they were intermediate steps. We didn't actually focus on reactions that were specifically targeted at pi bonds, double bonds or triple bonds. So that's what we're going to do in this chapter. We're going to get a running start on nonpolar pi bonds. And we're going to, in fact, it comes in two parts. First part is we're going to say what happens to a pi bond when you introduce a Bronsted acid into the mixture. How does that react? Well, as it turns out, pi bonds are particularly reactive. They behave as nucleophiles. In other words, they have excess electron available, and that comes from the pi bond itself for several reasons. One is that the pi electrons spend their time off axis from between the atoms. In other words, they're more exposed, so to speak. Now, that means that the pi bond is weaker than the sigma bond, but it also makes it more reactive. And if we look at where the pi bonds are in the molecular orbital format, they're higher energy than the sigma bonds. The bonding pies are higher energy than the sigma orbitals. And of course, the trend hasn't changed. I mean, not the trend, the convention hasn't changed. Whenever you have a single line, it's 2 electrons. So that second electron on the cyclohexene, I mean, that second line on the cyclohexene is a pi bond. And those are the electrons that are nucleophilic. So when we define what we're studying now as electrophilic addition, the electrophile is the acid. And it goes after those electrons on the pi bond. Example. So when we put hydrochloric acid in there with this single pi bond, we add hydrogen on one side and chlorine on the other. And the pi bond's gone now. And we call that addition product an adduct. That's short for addition product, ADD-UCT. Very inventive. We can do a similar thing with concentrated sulfuric acid, although they are slightly different in the way they behave and the selectivity that they exhibit. And we're going to look at that in a few minutes. So for this 2,3-dimethylbutene, when we add acid to that, sulfuric acid, remember when an acid dissociates, it does so 1 proton at a time. So what we have left over here is that species. That's the species that let me see. Wait a minute, H2SO4. Yeah, that's right. H. Okay. Oh, I see. Okay. What they've done is they put one of those oxygens on this side because the bonding that takes place is with that oxygen. It's not with the sulfur. That's why they wrote it that way. So that hydrogen sulfate ion is takes the place of the chloride in the previous example. All right, so here's the mechanism. It's actually two steps. The electrophilic addition step is when the hydrogen ion is removed from the acid, and it's attacked by the nucleophile. Nucleophile behaves just like it always has. When you have a double bond, you've got these electrons here, and they attack whatever the acid is. So they form a bond. This attack forms a bond here with a hydrogen. So now we have a hydrogen, and that bond's gone. But that also leaves us with a carbocation on the other side of the double bond. And then the second step is a coordination where you have now that you have this removed, now you've got that to deal with. And in this case, it's a chloride ion, but it could be anything. And now this attacks there. And it's a coordination step because the bonding electrons come from one side only. Remember, coordinated covalent bonds form, both electrons come from one species. You don't share one from one from the other. That's why they call it coordination. If you hadn't already picked that up. So that's the mechanism. Now, if we look at this in terms of an energy diagram, when the nucleophile, the pi bond, attacks the hydrogen, the energy of the reacting species goes up. We reach an activation state. In other words, we need that activation energy in order to get there. And because, if we go back to kinetics, right? When we study and trying to figure out which step in a series is the rate determining step, then the one that takes the most energy to get there, right? When we're looking at an energy diagram, say it's something just simple like that. Then if this is one step in the process, the higher this distance right here, the slower the reaction. Okay. And the slower reaction in a series is the rate determining step in any mechanism. Okay. So that's why this first step, the addition of the hydrogen across that double bond, that's the rate determining step. Then once we get to that height, now we've got an unstable carbocation. And it doesn't take much energy then to get to its activated state. So the second step, where you have the chloride attacking that carbocation, that is a very fast step. So the rate determining step is the first one. The coordination step is really, really fast. All right. So get a fresh start.
Our kinetics in this class or 102? I can't remember. It's 102, right?
Yes. Yes, we
But
did.
did we go over it in here too?
A little bit.
I felt like we
Yeah.
did.
Yeah, kinetics we introduced kinetics here. It doesn't sound the same in organic as it does in inorganic, as general chemistry. I think the organic chemists take liberties.
Yeah.
It just occurred to me today, organic chemists are more like glorified cooks than anything else.
Yeah.
They just go into the lab and cook stuff up and then try to explain it.
Yeah.
Now, they would probably take issue with me and say, no, we're bona fide scientists. Well, okay. You keep on believing that. What drives the reaction? in an electrophilic addition, in general terms, that is, reactions are energetically quite favorable. And we can see that on that previous slide, where we see the difference in the energies for the reactants is higher than it is for the products. So this is an exothermic, or I think they like to use exergonic. So we don't actually focus on whether it's heat. Exothermic means heat. Exergonic means we cover all types of energy. So it's energetically favorable. And another way to look at that is if we the difference between the bonds lost and the bonds gained, right? And we looked at that in terms in general chemistry in terms of writing the well, we may not have done that in 102. I could have done it in 101. When we look at how to determine the enthalpy for a reaction. And all you have to do is just add up the bonds broken, subtract the bonds that are formed, and you get a value. And you just have to identify the bonds. Like a carbon-carbon double bond has so much energy, and the single bond so much energy, and it's just a math problem. But when we look at it this way, we find that for the bonds broken, we have one pi bond and one sigma bond broken. But we gain two sigma bonds. Sigma bonds are stronger. So if you lose a pi bond and you gain a sigma bond, you've favored yourself energetically by forming two stronger bonds out of a weaker and a strong bond. I think that's the whole point of that slide. So which one of these will react faster? They both have a single double bond. Sounds funny. A single pi bond. But the difference is in the formation of the carbocation. So the fast step, the slow step, the rate determining step is going to be the electrophilic addition of the hydrogen across that bond. But when it does that, it forms a carbocation. So we know that the more stable the carbocation, the faster the reaction is going to proceed. So which one's going to have the more stable? Well, they're symmetrical, so it doesn't matter which one is attacked. If you attack, let's see, 1, 2, 3, 4, 5, 6. Let's see, it starts up. 2, 4, 2, 3, 4, 5, 6. And this is a double bond, okay? And we're comparing a cyclohexane with a couple of methyl groups on it. So this one has more carbons in it, but they each have one double bond. So which one's going to go faster? Well, it depends on which one forms the more stable carbocation. So if your hydrogen comes in here, if this attacks the hydrogen and it gives you a hydrogen here, now this is gone, and this is a carbocation. If the same thing happens down here, then the hydrogen comes in here, attacks there, and we get a carbocation there. So this is a secondary. This one is a tertiary. That one's more stable. That means this reaction is going to proceed faster. And that's on the next set of slides. But I just wanted to steal my own thunder.
So anything that's like, if there was a quaternary, it would be more stable.
Mm-hmm.
So we would choose that one over.
Yeah.
Okay.
Is it possible to develop a quaternary carbocation with a double bond? Because you'd have to have 3 carbons attached. Let's see, you have to have 4. 3 here, 2 there. You have to have 4. I don't know how you do that. You could have, yeah, you could have 4. You could have 4 carbons attached, but you couldn't have a double bond though.
Okay.
Yeah, because a carbon normally has 4 bonds of some kind, and if you had 4 carbons already there with single bonds, then there's no place to put the double bond.
Yeah.
Right. So quaternary is a non-starter.
Okay.
All right. So, protonation of A has a smaller energy barrier and is faster, making the overall reaction rate A faster than B. Now, since the energy barrier is lower, then the rate determining step is faster. So that first step, when we form the carbocation, it's more stable, which means it's at a lower energy, which means the energy barrier is lower. I wonder if that's on the next. Is that on the next slide? No, it's not. Okay. I thought we might have had a comparison. But if you look at it in terms of going up, like we have this one here, and then this one here, and it's like that. So we have this barrier for our rate determining step. But if this is for one of those, this is for the 1, 2, 3, 4, 5, 6 hexene, hex 3-ene. The hex 3-ene would be this one. And then the cyclohexene would be, let's make that dot line. It would take less energy to get to there. So the rate determining step requires less energy to get to that transition state because of the stability of the carbocation. All right. We're going to look at aromatics later and how they react. But this introduces the fact that aromatics like benzene do not react with acids. We will not get an electrophilic addition across any of those bonds because they're not really double bonds. I've mentioned this several times. I don't remember where, but they're the electrons are delocalized over all these bonds. We know that because the bond length here on each one of these is exactly the same. If this were the case, then this would be shorter than that one, right? But it's not. So that's why sometimes we draw it with a circle like that to show that those electrons are spread. That means that it's more stable in this format and the pi electrons are not available to react. So we don't get this type of reaction with aromatic compounds. And it just occurred to me, that would be one way. I mean, when we get to that chapter on aromatics, we're going to have some rules to follow to determine, is this compound aromatic or not? And we can do that. But I suspect that it's based upon physical evidence, right? If you can get a compound with double bonds to react with an acid, then it's probably not aromatic. But if it won't react, then it probably is aromatic. And does it obey the rules that you've set up? Yeah, okay, we're good. That's my way of thinking anyway. All right, so if we have this compound, 1,2-diphenylethene, then we know that because the aromatic parts of that molecule are not going to react, then we can add a hydrobromic acid across the double bond and brominate it effectively. And we know it won't affect the aromatic side ends at all. The phenyl ends won't react. We'll only get that reaction. 1 bromo, 2 phenyl, 2 phenyl ethyl benzene. Okay. Now we're going to look at what the Markovnikov rule. When we have an asymmetric double bond, right? That previous one, we had a symmetrical double bond where we had the same number of carbons on both sides and no other functional groups in the way. But if you have an asymmetrical like this propene, right? This is less substituted with carbons on this side, more substituted here. So which side does it attack? Well, it attacks so that you get the more stable carbocation. So let's look at it this way. Which would be the more stable carbocation? Be this one, wouldn't it? That's the secondary one. We wouldn't get one on this end because that'd be a primary. So that means that this attacks, let's see, we're using, hydrochloric acid. This attacks that one and forms the bond here.
Now, does it just leave or does the double bond kind of unfold?
Oh, the electrons form the bond with the hydrogen.
Oh.
Yeah. Right, they're out there in that space around the sigma bond, and they are going to attack or form the bond with hydrogen. They're right here now. So this bond here becomes this bond here. This goes from a pi bond to a sigma bond. And leaves behind that carbocation. So this is the Markovnikov carbocation. The anti-Markovnikov would be this one, with the hydrogen forming there. Now that does occur, but not in this case. Right? The incentive to form carbocation like that is stronger. Well, it's a major product. Let me put it that way. This forms the major product is this one, where you have the chloride here and the hydrogen there, and a minor product. So what we're saying is it does happen, but the major product is this one, because of the stability of the carbocation. So these are constitutional isomers of one another. In other words, they're not mirror images and they're not diastereomers. They have different, I forget the word, connectivity. They have different connectivity. Right. All right, so if we look at it in these terms, the more stable carbocation gives you a lower activation energy for the rate determining step. So that's why I put a blue box around that bottom one. And it corresponds to the blue line on the energy diagram. So that it takes less energy because the carbocation, the secondary carbocation, is more stable. So that one goes faster. So this is a kinetic control. This reaction is under kinetic control, and the one that occurs faster is the major product. So the 2-chloropropane is the major product, and the 1-chloropropane is the minor product.
So the minor's slower?
Yes, it's slower because it takes more energy to get to the activated state. And since that's the rate determining step, it's slower, and the kinetic advantage is toward the secondary carbocation. And from then on, the amount of energy required to go from the intermediate step to the activated state with the formation of the chloride bond is about the same. So that's not an issue. Okay. So this is Markovnikov's rule. And it simply says that he was Russian. The addition of a hydrogen to an alkene favors the product in which the proton adds to the carbon that is initially bonded to the greatest number of hydrogen atoms. It does that so that you can get the more stable carbocation. You can look at it either way, right? You can add your hydrogen to where it has more neighbors, more hydrogen neighbors, or you can think of it in terms of the carbocation. Actually, the carbocation explanation is why this Markovnikov addition works the way it does. Okay, let's see. There's another example up there, but it's based upon the same premise. How about this one? Indene. Predict the major product when indene is treated with HCl. All right, so we know it's not going to add to this one. It's like that, yeah. This is known as a fused ring. When two rings share two carbons, those rings are fused, not just connected through a single. So let's see, this one's like that, and that one's aromatic. Okay, so we know the reaction is going to occur here. Well, which one is more substituted? Which one, actually, not just which carbocation is more substituted, but which one is more stable. There are other factors that can stabilize a carbocation. And one of those factors is I have trouble with my words today. Is the resonance stabilized? Okay. So if the hydrogen were to add to this one, right, they're both equally substituted, then you would get a carbocation out here, which would be a secondary, right? It would be like that. But if it forms over here, then we can resonance stabilize it with this aromatic ring. And what will happen is, well, it takes advantage of the stability of these delocalized electrons, stabilize that one. So even though it's a secondary, it's more stable by its proximity to this aromatic ring.
Why won't we choose the fused area since it's tertiary?
Oh, put it here.
Yeah.
Because it won't form a carbocation here. That's not a good explanation, we think. I had never thought of that. Like, if it actually formed here, then why doesn't it? Why don't we get a hydride shift, right? Because there's a hydrogen here. Why don't we get the hydrogen over here and the carbocation move over there? I'm sure there's a good explanation. Just I can't think of one.
That's okay. I probably wouldn't understand anyways.
It doesn't happen.
Yeah.
Because I think if we put the carbocation here, then we would destroy the aromaticity.
Oh.
Yeah, that understanding will come later when we look at rules governing what is an aromatic compound. Because some aromatics are charged species. And if we charge one that's already aromatic, I suspect it will destroy that aromatic character. That's my explanation, and I'm sticking to it until somebody comes along with better. So our major product is going to have the chloride. The chloride bond will form closest to the aromatic ring. Now, it could be no, it's going to be right there. Yeah. So that bottom one is going to be the major product. Now, that doesn't mean the top one doesn't form, but the major product will be on the bottom. I mentioned rearrangements. You can get rearrangements. Excuse me. And when you do that, in this case, actually, you get an equal amount of these two isomers. Yeah. Of course, the hydrogen is going to be where we expect it to be. on the more hydrogen substituted carbon. But why then would the chloride not be over there where the hydrogen is, but in fact it would be on the more carbon substituted? The carbocation would have formed. Let's see, let's draw this out. There, there, and there. So when we get the Bronsted acid approaching, then this is going to attack that. And the hydrogen is going to attach here. And that leaves us with a secondary carbocation. But it would be more stable over here. I mean, we can still do that and form the bond here. But we can also get a hydride shift. This hydrogen and shift over here. And that leaves a positive over here. And now another one of those can attack here, and we can get a mixture of those two, because the hydride shift occurs with the intent of stabilizing the carbocation. We looked at that several chapters ago. We can get hydride shifts, we can get methyl shifts also to stabilize these carbocations. And if it's energetically favorable to do that, then it will shift and we get the carbocation over here, and then the attack occurs there. And since this is the slow step, we've got time to do this shift before we experience another attack. Right. So this shift happens very quickly. And that would be my only explanation for why you get an equal mixture of the two isomers. Carbocation rearrangement. Okay. Oh, all right. Here's the mechanism. So I got that part right. When we first formed the first one, then actually I didn't write this correctly. Well, yeah, I did. Where electrons are moving over here, and the bond forms here. So that means this positive is quenched, and that leaves behind a positive there. Okay. So now we have that other carbocation that will also form a coordination bond with chlorine. So the 1,2 hydride shift can account for the formation of these two products. All right. Let's see. Are we going to have the same situation with this one? Yeah, I bet we are. Yeah, I bet we're going to have we can't have a hydride shift because there's no hydrogen there, right? The carbon on that cyclohexane already has 4 bonds, right? 1, 2, 3, 4. So we can't shift to hydride. So what we have to do instead is when I think it's hydrogen bromide this time, it did. So when this attacks there and we form the bond with hydrogen here, then get a carbocation here, and the bromine can attack here, right? We can get an attack there, but we can also get a methyl shift. My guess is that the methyl shift happens slower.
Wait, won't we take the bromine off first and then add it?
Well, yeah.
You're just skipping a step.
Yeah, I'm skipping.
Okay.
Okay. All right. So now that's gone because it's over here.
Yeah.
And this is available to attack that carbocation. But we can also get a methyl shift where the electrons here attack there and form a bond with the methyl. And now we have a carbocation here. And then we can attack there. So we should have 2 products possible. A 1,2 methyl shift will also, let's see. Yeah, it would have to be a methyl shift to do that. So electrophilic addition there, okay, 1,2 methyl shift, right? And then coordination. Right, what's the question? Detail mechanism and predict the major product of this reaction. Well, I guess that would be a major product because the carbocation is more stable. But based upon our previous example, right, here, we got equal amounts, right? There wasn't a major product. Maybe it has to do with the fact that the carbocation here now is tertiary rather than secondary. Well, it was the tertiary and the other one too. Is there a stabilizing effect from a cyclic? Maybe their delocalization here is more effective. That would speed this up and give you a major, actually, it may shift the percentages toward the more stable carbocation. That's the only explanation I can think of. The author is saying that the major product is the one where bromine attaches there. Okay. Stereochemistry. So during these reactions, if you can form a carbocation, then the coordination step can attack from the front or the back, because now carbocations are planar. Remember? Yeah. So you can attack from the front or back, and you should get a mixture of R's and S's at that chiral center. Right? We don't have a chiral center when there's a double bond being attacked. That's not a chiral center. But when you open up that double bond and make the carbocation, now you can attack from the front or the back, and all you need is 4 different groups around a carbon, and that will be a chiral center. So if you have a carbon and maybe you have a hydrogen and you have 4 other things, then you have a chiral center. If any 2 of these are the same as one another, like if this one is the same as that one, then that's not chiral. They have to all be different. And that's exactly what we've done here. All right, we've got at that carbon, we have a bromine, we have a hydrogen, we have a phenyl group on one side, and we have a benzyl group on the other. All right, so there are 4 different groups around that carbon. That's a chiral center. So we get in this case, a racemic mixture. Racemic mixture means that we have equal amounts of both of them. And that's largely due to the fact that that carbocation is the only one that's available. We can't stabilize it any other way by a hydride shift or a methyl shift. So we get equal front side and back side attack. All right. In this case, let's see. In this case, we have 2 carbons, right? It's symmetrical to start with, but let's look at the mechanism. That'll make more sense, because what they're saying is we have 4 stereoisomers here. Oh, yeah, sure. Yeah, we created 2 chiral centers, one where the hydrogen attached and the other one where the bromine attached. Yeah, those are 2 chiral centers. So that means we could get a front side or a back side attack for the coordination, for the electrophilic addition. Right, that's the first step. We could get the hydrogen adding on the front side, or we could get the hydrogen adding on the back side. That would give us an R, a difference R, an S there. And then once we have the carbocation, we can attack from the front or the back there, and gives us another RS combination. So we have 4 combinations there. We could have both R's, we have both S's, or a flip-flop between R and S. I mean, does that make sense? Okay.
Yeah.
All right. So up to now, we've used strong acids, hydrochloric acid, sulfuric acid, hydrobromic acid, which hydrobromic acid is even stronger than hydrochloric acid. It takes a strong acid to undergo electrophilic attack, electrophilic addition. If we try to use a weak acid, the reaction won't go. I mean, we can't add across that double bond with just water, because water is a weak acid. We know water is a weak acid. Why? Because the KW is 10 to the minus 14. Right? That's a very weak acid. But if we can protonate that, actually, it makes more sense to do it this way, to do it the actual way that it occurred in pure water. We get a hydronium ion. There we go. So if let's just arbitrarily say that's the acid and that's the base. That makes this the conjugate acid of this base, and this is the conjugate base. It's acid. These are both weak. So this weak acid makes a strong base, and this weak base makes a strong acid. So these are both strong. If we can make that strong acid in solution, then we can get an addition across that bond. Right? We can give up the proton here. Let's see. So H plus. We can use one of these protons on the double bond. Right? So it can be like that, form that, and that gives you water. And then in that same solution, you should have some hydroxyls. That may not be the mechanism. Let me not muddy the waters. But I'm pretty sure this is the first step. But if we use an acid like sulfuric acid, then we can protonate one of these water molecules and make a strong acid. And if there's just enough acid in there to make this, right, we don't want a lot of sulfuric acid because we don't want to add the hydrogen sulfate across the bond. We want to add the hydroxyl and make an alcohol. So this is known as an acid catalyzed hydration. All right, and it's fairly efficient actually. All right, let's see what the mechanism looks like. Okay, I got that part right. The first part, the electrophilic addition of the hydrogen, forms the carbocation, okay? Then the carbocation attacks the water. All right, so I was heading the wrong direction. So once we've got the carbocation here with our hydrogen attached, then the water molecule, like that, and it attacks the carbocation, those two, and attaches that water molecule to the carbocation position. Now we deprotonate it. So what we have now left over is this hydrogen as oxygen and the two hydrogens. But that means it's a positive charge. And then these electrons go here, and this proton goes back to water. Just like that. So it forms the bond with water. It reconstitutes this acid. That's why they call it acid catalyzed, because after the reaction is completely done, you've reconstituted the acid that was used. It goes in one side, comes out the other. So that's a catalyst. And now we have an alkoxylation. Well, actually, we have a hydration. We've hydrated that bond with water. But if we have, instead of water, say we have an alkoxide, like that, we get a similar reaction. And instead of hydroxyl, we get an ether. Okay. All you have to do is substitute an R group for the OH. So we have an OH. Let's see. Here, instead of like this, that, change one of those to an R. And you get the same type of reaction. Only now you haven't extended the carbon skeleton, but you've created an ether. All right. Now, why do we not use hydrogen chloride as an acid catalyst? Okay. It has to do with, let's see, SO3, right? Yep. It has to do with the way this double bond attacks here, right? And that leaves us with SO H, missing the hydrogen. There we go. HSO3, HSO4 minus. There we go. This is a weak base. It's not a strong nucleophile either. Okay. Whereas this one is not a strong base either. Actually, this is a stronger base than this one, but this is a very, very strong nucleophile, which means that if you put this in aqueous solution and think you're going to add hydrogen on one side and hydroxyl on the other, make an alcohol, you won't do it, because this is much a stronger nucleophile than hydroxyl, and it will add instead of the hydroxyl. So that's why hydrochloric acid is not used as an acid catalyst. You need something that produces a weaker nucleophile. And I suspect this is a weaker nucleophile, even though it's got all these oxygens on it, with their electrons, it has spread them over a larger area. The electron density is much lower in this one than it is in this one, because this is smaller. All right. So let's see. I should have gone straight to that. Hydrogen chloride is another strong acid that dissociates in water, but HCl does not catalyze the hydration. The chloride is a strong nucleophile, much stronger than HSO4 minus. Okay, I already told you that. So in this case, you get a chloride addition on it rather than a hydroxyl addition. Now, what happens when you encounter an alkyne? We have two pi bonds. What you get is a double chlorination on the same carbon, or a double halogenation of the same carbon. And I think it all has to do with the stability of the carbocation. Because you're going to form carbocation twice in this reaction because you have two pi bonds. So They never did define what geminal dihalide is. Geminal. Geminal means giving birth to, doesn't it?
I'm looking it up right now.
Yeah. Geminal di something.
It says when both the halogen atoms are present on the same carbon atom of the chain.
Well, that doesn't tell me anything. I need the etymology.
Mm.
Did you ever go to etymology online?
Mhm.
Yeah, go to that website and plug in a word, and it'll tell you what each part of the word means and where it came from.
No. Give me geminate, not geminal. But it means geminate means duplicated, found in pairs
Okay.
or twin.
Oh, yeah. Oh, they're going from the Latin.
Yeah.
Right. Gemini is twins, the twins.
Yeah.
Right.
Oh, Gemini. Yeah.
Yeah.
Well.
So that's where it comes from. All right, now it makes sense. All right. So if we look at the mechanism, we get the first step, the electrophilic addition of the hydrogen is the same as before, only now we've got a vanillic carbocation. A vanilla, well, it's not, well, they don't call it carbocation, a vanillic cation. So the carbon has a positive charge, a formal positive charge, but you still have a double bond left over. So the chloride is added to that carbocation as before, coordination step. And then another hydrogen chloride molecule is attacked by the remaining pi bond. And which carbocation is more stable? The more substituted one. Right. And in this case, it's not just more substituted as carbons, but it's got a chloride on there too. Actually, that would make it less stable. All right. Chloride has an electron withdrawing effect, so it'd make it more positive. All right. So my guess is the second electrophilic addition happens slower than the first one. So I would be willing to bet that the second electrophilic addition is the rate determining step. Simply because that chloride destabilizes. Well, now they're making a liar out of me. There's resonance involved. I forget what I just said. This resonance stabilization with the chloride will stabilize that carbocation. So my guess is the kinetics were worked out for this experiment, and then they had to explain why the second step was faster than the first one. And that's their explanation, resonance stabilization. Okay, but the reaction still occurs with the carbocation on the carbon, the carbocation on the carbon and not the positive on the chloride. Right, so you wouldn't attach another chlorine to that to that the chloride positive. And that's supposedly the explanation why you get a geminal halide. Right. So for this reaction where you have propine, we're going to add bromine across on the number 2 carbon. It's more stable. Now let's see. If we don't have resonance, what's the situation? Well, we find that we still get the geminal halide, 2,2-dibromopropane. We still get the geminal halide, but it happens by a different mechanism because we don't have resonance stabilization available to us. So here's what the author says happens. we form a pi complex of the acid with the alkyne. And the bromide ion then attacks the more substituted carbon and leaves the hydrogen behind. Then that bromide separates, the top bromide separates, and now it's able to attack again. I think it didn't tell us that it attacked the second time. Well, it has to attack another hydrogen bromide. That's the only explanation. All right. So we're back to acid catalyzing, this time an alkyne. So what does that produce? Well, instead of an alcohol, it produces a ketone. Yeah, acid catalyzed hydration of an alkyne produces a ketone, not an alcohol. All right, so I suspect that the first step is the same. Right, the electrophilic attack would be the same. And you would form the more stable carbocation, and then the water acts as a nucleophile, and we attach water to that carbon where the carbocation used to be. Then we deprotonate it. That leaves the hydroxide. That leaves an enol. An enol is less stable than a ketone. So we get a keto, an enol. Actually, they say it keto-enol tautomerization. We just move the double bond. It's essentially a hydride shift from the oxygen over to the carbon. Have we discussed tautomerization yet?
I think so.
Yeah. Okay. This is a process of tautomerization. The keto form is more stable. So as soon as you form that enol, it immediately tautomerizes. And it does so actually through 2 steps. Let's see, are they going to show us those steps? Yeah. Okay. Let's see. Actually, I think I added some comments. Get this out of the way because I'm pretty sure they're going to come up here. All right. So the product in step 3 is an enol, right? We've got that far on the previous slide. Under these acidic conditions, the enol rapidly tautomerizes to the more stable keto in 2 steps, which are both proton transfers. So how would that happen? Proton transfers. My guess is that the proton transfers are not intramolecular, they're intermolecular. They happen with water as an intermediate. Yeah, I think that's what happens. So that hydrogen on the alcohol is transferred to water. And then let's see if I can draw that out. I don't know if I can, but I'll try. So if we have this one to start with, like that, and then we have this one and this one here, there, and with the hydrogen. So hydrogen's there. Okay, so the first step would be for water to deprotonate that. What would that leave behind? That would leave behind. Let's see, I put my double bonds in the wrong place, I think. Anyway, it would leave behind this positively charged, right? No, negative. Because the electrons that were in this bond, actually, I need to draw it this way. That bond, then those are transferred here, so that gives us an octet there. But it's a negative charge now. Okay. So that one proton transfer, then these electrons here, let's see. Yeah, these electrons here go there. So that gives us, and that gives you double bond oxygen here, but it leaves you with a positive charge there. So then, this positive charge thing here, no, that doesn't work. All right. I obviously don't know what I'm doing. I got as far as the double bond, now I don't know what to do. We need a proton transfer to this one. Maybe it doesn't use that one, it uses this. Yeah, it uses this one. So in order to attack this one, we'd have to go from here and form this like that. That doesn't work either. Never mind. I don't know what I'm doing now. After I get past that step, I'm lost. It just says 2 proton transfers. So important. The takeaways here are the product is a ketone, not an aldehyde. An aldehyde would put the carbonyl on the end carbon, and that's not where it goes because that's not where the original carbocation was located. We run the reaction at very high temperatures so that the addition of hydrogen ions in the first step of the mechanism produces a vinylic carbocation intermediate. This intermediate is rather unstable. First step, therefore, has a very large energy barrier when hydronium is the acid, which can be surmounted with large amounts of added heat. All right, what are we saying there? Remember, if it's large heat, that gives you a not a kinetic controlled rate mechanism step, but an equilibrium control step. Because if you have a lot of heat, then when you form this unstable compound, you got enough energy to go back the other direction. So you go both ways, and that stabilizes the process so that you maintain the reactive species on the other side. Otherwise, it's too unstable at lower temperatures. You need the higher temperatures to switch from a kinetic to a equilibrium controlled or thermodynamically controlled. So how do we increase the yield of an acid catalyzed hydration? Well, we need to increase the strength of the catalyst. We need something stronger than sulfuric acid. And this, let's see. What's that called? Come on. There we go. Trifluoromethane sulfonic acid is stronger than sulfuric acid. And of course, we put it in trifluoroethanol because of the common molecular formula. And that gives us 100% phenylethanone. Well, 100% theoretically, right? I would never expect to get 100% from a reaction.
What is the odds of getting 100%? Like very low.
Very low. Yes. You're always going to have some, I mean, even if it's not traceable to side reactions or equilibrium reversals or anything like that, or side reactions would be undesirable products. Even if it's not that, when you isolate the product from the rest of the mixture, you're gonna lose some. You never have 100% recovery. So that's theoretical. So what if we have a double diene, or not a double diene, that's redundant. What if we have a diene, right? We can have two possibilities. We could have a 1,2 addition or a 1,4 addition. Okay. Are we going to add to the number 1 ene or the number 3 ene? Right, we have a butyl 1,3-diene. Right, so we get two products. We get one where obviously it added to the 3, and the other one added to the 1. And when that happens, when we add to the 3, we still have the 1 double bond. But when we add to the 1, then we shift the double bond from 3,4 to 2,3. All right, so we do get both products. So when we add the hydrogen and chlorine across the same bond, we get a 1,2 addition. Right. So I would expect, let's see, the one on the left. Yeah, the one on the left would be a 1,2 addition. Because the first one, the one on the right, since it's symmetrical, it doesn't matter which side you count from. The one on the right, you would add the hydrogen to the first carbon, and then the carbocation would add the chloride on the second one. That's a 1,2 addition. If, on the other hand, you add the Markovnikov is going to add the electrophilic addition of the hydrogen is going to be on the 1 in both cases. Right? Because the hydrogen substituted carbon is on the end. And since the double bonds are at the end of the molecule, we can start from either end, call it 1. Doesn't matter. So the first step is the same for both of them. Right? You're going to add the hydrogen and form the carbocation. So the first step is forming the secondary carbocation, where the 3-chlorobutadiene locates the chlorine. Now we have to explain why then do we end up with chloride on the number 4 position. Instead of 1,2 addition, we get a 1,4 addition. All right, so that's the trick. So what we get is a resonance stabilized carbocation. And that makes the carbon with the positive charge available in 2 different locations. This resonance hybrid gives us a secondary carbon, and it gives us a primary carbocation. So we get both 1,2 addition and 1,4 addition products out of this reaction. Produced out of the same intermediate. The intermediate here is the resonance hybrid, that one at the bottom. That's considered the intermediate, and we get both carbocations out of that. Now, which one is favored? That's my question. Okay, they're going to explain the mechanism to us. All right, so the first step is the electrophilic addition of the hydrogen. Right? and it's not going to put the carbocation on the number one carbon. So we can't explain it that way. So, actually, we're going into control. How do we control which one is produced, the 1,2 or the 1,4? Because both are possible. So it's a temperature thing. Under high temperatures, we get the thermodynamic control, like I mentioned earlier, where you establish equilibrium. Because with high temperatures, you have enough energy to go backwards. That's what that energy diagram is showing us. Right, that tall red arrow. At high temperatures, you have enough energy so that you can go backwards. Thermodynamic control means the one that is the lowest in energy is favored. The major product has lower energy. So under thermodynamic control, you're going to get a 1,2 addition because the carbocation in the 1,2 addition gives us the more stable carbocation. Under low temperatures, you're going to get kinetic control. Which one happens faster? So the electrophilic addition to, excuse me, to the conjugated diene is irreversible at low temperatures. So the one that is produced more rapidly is the one that is more stable. You know, I think I got those turned around. One second. The thermodynamic product of the electrophilic addition is a conjugated diene, is generally the one remaining. Carbon-carbon double bond is the most highly alkyl substituted. So in the 1,2 adduct, the alkene is monosubstituted. So for thermodynamic control, we're going to get a 1,4 adduct because the alkene is disubstituted. That's the more stable alkene. So the 1,4 is under thermodynamic control. That's what they're saying. Let's go back to the energy diagram so I can make sense of this. The 1,4 at high temperature is under thermodynamic control. That doesn't help me. This makes more sense. Yeah. The carbon-carbon bond that's more substituted is more stable. So we find the 1,4 adduct is more stable at high temperatures, and the 1,2 is more stable at low temperature. It's under kinetic control. So at high temperatures, we're going to get 75% of the 1,4 adduct. And then at, let's see, 20 degrees is considered high temperature. Compared to what, right? I guess compared to absolute zero. So if we carry it out at room temperature, we're going to get the predominance is going to be 1,4 adduct. But if we cool it down, say we ice it down, then we're going to get the 1,2 substituted. Major product at cold temperature then is the 1,2 substituted. And the ene that's left over is less stable. Ooh, minus 80 degrees. Yeah. I don't even know if you can get that cold with dry ice.
How cold is dry ice?
It's freezing point is minus 78.5 degrees C. So it's pretty close.
Yeah.
And it might work. Maybe they're just saying minus 80 is rounded off. It's probably dry ice that they're using. Probably take longer too.
Can you like enhance, well, I don't really know how to say this. Can you like do something to dry ice to make it react more or make it colder?
Yeah, probably could. How would you do that? I know how to do it with water ice. You just throw salt on it, right?
Yeah.
Let's see, could we do that with dry ice? Dry ice, though, it sublimates. What if we added water to dry ice? Look it up. How to make dry ice colder. Well, we've got time to digress.
I mean, it's just telling us how to make it.
Dry ice and isopropanol. Okay. Does it say what temperature it is?
No, I think that was how to make it.
Okay.
Well, how to make cryogenic fluid from dry ice and rubbing it over.
Well, that would be colder.
It doesn't say, it's just how to use dry ice.
Because solid carbon dioxide is not cryogenic. Liquid carbon dioxide is not cryogenic.
Yeah.
Okay.
Not seeing it anywhere.
Let's try this one. This one's mixture with ethanol. After you mix the ice bath, it will stay around -6. No. That doesn't make it any colder. I don't know. That's a question for the ages.
Trying to look it up, but it keeps on telling me put it in the cooler. I was like, well, that's definitely not it.
Right there. Their artificial intelligence algorithm that just isn't up to the challenge.
Yeah.
Okay. So, all right, this 1,2 adduct is kinetic product because of where the chloride is located after completion of the first step of the mechanism. All right. So if the first step is and this is at lower temperature. We're at -80 degrees. Then we get that first electrophilic addition of the hydrogen and forms the resonance stabilized carbocation. That's what that middle picture is. It's resonance stabilized. The proximity of the chloride that's formed when the hydrogen breaks away from the chlorine, the proximity puts it closer to the number 2 position at those temperatures. I mean, it's not, it's a lot colder, so it's not moving as fast, right? The kinetic energy is not there. So since it's colder, it's closer to the number 2 carbon, and that's where it attacks. That's the explanation for the kinetic control of the product.
I asked AI and they said add liquid nitrogen, a vacuum chamber, or mixing it with a substance with a lower freezing point.
Substance with a lower freezing point.
Mhm.
Oh, what would be a lower freezing point than carbon dioxide?
Substances that freeze at lower temperatures than dry ice, like certain alcohols, can be used in a mixture to create a colder environment.
Okay.
For instance, a mixture of dry ice and acetone or ethanol can create a slurry with a temperature lower than the sublimation point of dry ice. However, this should be done with caution and proper ventilation due to flammability and toxicity of the solvent.
Oh, okay. Okay. Now we know.
Yeah.
Use what, isopropanol or acetone?
Yeah, it's saying acetone or ethanol. So,
Ethanol. Okay.
yeah, or liquid nitrogen, or you could use a vacuum chamber.
Well, just, I mean, if you really want to get it cold, minus 195 degrees Celsius is liquid nitrogen.
Yeah,
But if you conducted the reaction in the vapor above the liquid nitrogen, that wouldn't be as cold. It might be the ideal temperature then.
yeah,
You'd have to measure it. It's whatever you have available, I guess.
yeah. This says, however, this requires specialized equipment and safety precautions. For the liquid nitrogen.
Yeah. We used to use liquid nitrogen and carbon dioxide.
Isn't that what they
dries.
spray on like warts, or like before you get like a cortisone shot, they spray liquid
Yes.
nitrogen? Yeah. Okay.
Yes. Yeah, it burns them off by going the opposite direction of temperature.
Yeah.
It freezes them.
Yeah.
It kills the tissue. Yeah.
But don't they do that when you get like a cortisone shot too, like spray it? No,
They don't use liquid nitrogen for that.
Well,
They use like a fluorocarbon.
okay, I thought I saw
Freon.
nitrogen on it because I had one put in my knee one time and I thought it was nitrogen.
Yeah, was it? Well, maybe they are
I
using
don't
it.
know if it was, but I think it was. Maybe I'm thinking back to when
You
I had a
can.
wart froze off.
I mean, if you don't hit it for very long, you
Yeah,
just want to numb it. You don't want to kill it.
yeah, that's what it was basically doing, just like get it numb, I guess.
Uh-huh. Yeah. It's safer to use a fluorocarbon.
Yeah, and I mean, it might have been that.
Yeah. But if they normally do dermatology at that practice, then they may have liquid nitrogen available.
Yeah.
And they just use it because it's there. But you have to be careful with it.
Yeah.
Now I can pour liquid nitrogen into my hand and it won't burn as long as I keep it moving.
Yeah.
It's called the Leidenfrost effect. In other words, it vaporizes so fast because the temperature difference between there's enough heat from my hand to vaporize the nitrogen, and it forms a vapor layer between the liquid nitrogen in my hand. That's an insulator. So as long as I keep it moving, if I stop and let it sit there, it'll burn a hole in my hand.
Yeah.
But all you have to do is just like that, it's gone.
Yeah. That's cool though.
You can see that Leidenfrost effect if you heat up an iron skillet, get it really hot, and then dribble some water in it, and you'll see the beads dance.
Yeah.
It's the
Yeah,
same,
there's
same
something.
effect.
Okay.
It's a temperature differential.
Yeah. That's how they say your stainless steel pan is ready to not put it on unless the water beads up. Because if you don't let it bead up, it'll stick. Because it from where the porosities, they close up when it heats up. So that's why it beads up, and then if not, the porosities are open. So that's why things stick.
Okay.
Just a little cooking lesson there.
Culinary arts, yeah.
Yeah.
I wonder if they teach that in the. I'll have to mention that to our director of culinary arts.
Yeah.
I forget his name. I haven't known him that long. Okay. So this explains the 1,2 adduct, which forms faster. Which means, in other words, if that chloride had enough energy to get to the other end of the molecule, we could also form a 1,4 adduct. But at low temperatures, that chloride is going to attack the closest positive charge it can find. And that's the number 2 position. Okay. All right. So now we're approaching this problem from a synthesis, a retrosynthesis approach. So if we want to have a brominated diphenyl compound, then we want to start with a a double bond. Then we can easily brominate that. If we want to form an alcohol or an ether, then we either want to add water to make an alcohol across a double bond, or we want to add an alcohol itself to form the ether. So either way will work. Right. Here are some other retrosynthetic analyses. Right, there's your geminal dihalide. Right? There's your formation of a ketone. And there's the alkyl halide. We're not going to spend a lot of time on that. All right. Suppose we want to go from this this bromide to the cyanide. All right. How would we do that? Well, let me see. It's probably going to take several steps. If we want to end up with a cyanide and we're going retroactively to the bromide, then that's going to be an SN2 substitution reaction. We're going to attack it with the cyanide, and the bromide would be the leading group. So that would be that one step. How do you get to the bromide? Well, if we brominate a double bond, we can end up with the proper bromide, because the bromine, actually, are we going to get the bromide we expect? Right? Yes. Right. We're not going to get a carbocation on the aromatic ring. So yes, that would work. So we're working backwards now. So we've got the, let's see, what is that called? My mind's gone blank. So in order to form that aromatic ene, we can work from a bromide. Yeah. So if we go from the bromide and now we're going to make a double bond, we need to attack it with an alkyl anion. That's a tertiary butyl alkylide ion, and that will form the double bond. Then we can brominate it the way we want, and then we can substitute the cyanide. So that would be your synthetic pathway. All right. There are a few slides here on terpenes, terpenoids, and the reason for that is because they are so important in nature. We can trace the structure of many biological molecules, biochemicals, by analyzing its, breaking it down into isoprene subunits. Isoprene is 5 carbons. One, two, three, four, 5. Right. That's an isoprene unit. Isoprene itself is a double bond here and a double bond there. So 2-methyl-but 1,3-diene is isoprene. But you can see 5 carbon subunits in lots of molecules. Right, here's some examples. Pine resin has this one, right? And we've got the orange backgrounds to show you where the isoprene subunits are. Limonene, right, from lemons. That's what limonene looks like. Zingiberine, ginger oil. That's a more complicated one. Now these, we can name them, right? A monoterpene has let's see. We're going to do that in a minute. Sembrine corals, I don't know about that one. Squalene. Squalene is very useful compound. 1, 2, 3, 4, 5, 6. 6 isoprene subunits. Squalene, and squalene is like a starting material for several compounds in biochemistry. And the cell makes stuff from them. It may take them and actually form ring structures, or it may form aromatic ring structures. I think squalene is starting material for several of the hormones, like estrogen and testosterone, for instance. Okay, here we have the naming convention. If we look at the number of pairs of isoprene units, a monoterpene is 1 isoprene unit. A sesquiterpene is still 1 isoprene unit, but there's a difference. The number of isoprene units, do we have, all right, okay, let me see if I can make sense of this thing. A monoterpene, oh, the number of pairs of isoprene units. So that means you have 2 isoprene subunits. Okay, 10 carbons. Sesquiterpene has 1 pair, or number of pairs of isoprene units. That doesn't make sense. That would be 1.5, right? 1.5 isoprene units, not 3. 2.5. If we have 1 pair, we have 1 complete pair and then 1 extra one. I guess that's what sesqui means. But sesquicentennial, that's 150 years, isn't it? Yeah, I think sesquicentennial is 150 years. So you go 1.5. So we have 1.5. So 2.5 of 2 is 1. So we have 3 isoprene units, but we only have 1 complete pair. A diterpene has 2 pairs or 4. A triterpene has 3 or 6 units. All right. Enough of that. These are other terpenoids are derived from terpene subunits, but oid means similar to, right? Like humanoid, similar to humans, or android, similar to man. So it's a little more difficult with the terpenoids to find out where those subunits are, right? Geraniol looks like it's fairly simple. Right, geraniol's got 1, 2, 3, 4, 5, 6, 7, 8 carbon backbone. 3, 4, 5, 6, 7, 8. Right? And then we have hydroxyl on the end. And on this one, we have there, and then we go over here and here, like that, right? Yeah, that's geraniol. So if we start from this end, we have 1, 2, 3, 4, 5. Right? And then 1, 2, 3, 4, 5. So you have 2. This is a monoterpenoid. We have 2. We have 1 pair of terpenes, or 2 terpene subunits, 2 isoprene subunits.
So I guess the retinol is different from retinol.
Yeah, Al means aldehyde.
Yeah.
Right.
And the all means.
If it was an all, there would be an OH out there on the end where the O is located. And that changes its activity too. You change the functional group and you change its activity. And that may be what happens in your eyes when it's exposed to light. It may change from one to the other.
Yeah.
One thing that, let's see, I don't want to draw the whole molecule, but I do want to say, right, if you have an alkane like this, and you have a double bond here, and you skip one, double bond here, skip one, double bond there. We're going to talk about this in more detail later in chapter on conjugation. But this is a conjugated bond system. And what that does is when you have a sigma and a sigma pi and a sigma and a sigma pi, you get delocalization of electron density over that whole system. And that stabilizes it. It lowers the amount of energy that it contains. So that's why when you get a molecule, well, I'm jumping ahead. Let's talk about it later when we get to spectroscopy. That's a conjugated system. I studied that a little bit as when I was working on crawfish processing waste. Crawfish have that red, like lobsters, they have that red color. It's a and it's red because of this conjugated system. Lowers the energy, so they absorb light in a different range and give you that red pigment.
So is that why, like, when you cook them, they get like the heat causes them to get a deeper red or like a brighter
Yeah,
red?
what happens is they are bound to other molecules in the shell, and when you heat it, you break those bonds, and that frees them to absorb light in a different range.
Okay. It's crazy to think of things that way, though. Like, you don't just, I don't know. Like, you don't think there's an explanation for it, but there always is.
No, you'd rather just cook them and eat them, right?
Yeah. Oh, for sure. I love crawdads.
Yeah, I lived for 13 years in Baton Rouge.
Yeah.
And I could hardly wait till the crawfish season opened up, because the first batches were the best and the cheapest.
Yeah.
And now I don't think I could afford it anymore.
Yeah. Yeah, no, everything seafood or anything water down is so expensive.
Now, we're not going to spend a whole lot of time on this one, except to say, in the cell, one of the ways to activate enzymes or transfer energy is to attach phosphate groups. So if you want a molecule to be active and available for reaction, you want to add energy to it, then you'll add a phosphate, right? So everybody's heard about ATP, adenosine triphosphate. That's an energy carrying molecule for the cells. And when it gets to where it's going, energy is extracted from it by removing one of those phosphate groups. So you make ADP, and you can actually get some more energy out of removing the second one, make AMP. And you can make a cyclic compound out of AMP, and that's a messenger molecule. So we won't go there. But it takes several steps to get there, right? We start with acetic acid in this case, and we make mevalonic acid out of that. And then we add phosphate groups. And isopentenyl diphosphate is, let's see, I'm covering something up here. Yeah, okay. Is available for further reactions. All right. I'm not going to dwell on this. Let's just see what the comments are. Terpene biosynthesis involves reactions that proceed through carbocation intermediates. Okay, I'll buy that. This carbocation chemistry is made possible by two features of isopentenyl diphosphate. One feature, it has carbon-carbon double bonds. Okay. And the diphosphate groups, which are good leaving groups. Okay, so if we're looking at this from an organic chemist standpoint, then we're focused on the leaving groups. And the diphosphate is a good leaving group. So that gives you the opportunity to make the double bond. But these reactions are enzyme mediated. So the enzyme provides an alternate pathway that lowers the activation energy. just like any other catalyst. Only with an enzyme, the enzyme is very specific. It'll catalyze only one type of reaction, and usually in only one direction. So enzymes give you more control, and that's what the cell needs. It needs to control reactions. Okay, I'm going to skip over that. Synthesis of neryl diphosphate. All right. I'm not going to spend a lot of time on this one either. If I accidentally put a homework problem in there that dwells on this, just do your best. Since it's extra credit, then you're not going to lose anything. These are natural product. I think the whole point behind this series of slides is to just make the connection between these organic steps, in particular the electrophilic addition that we just learned about. And it seems to be a very commonly used reaction mechanism in natural processes. That's it.